All right, now for introductions. Now I seem like I'm really loud. All right, so who wants to go first?
I'm going first.
You're going first. All right, so first we'll have Dr. Cushing, Paula Cushing, from the Denver Museum of Nature and Science. I hate to admit, but I don't remember all your education and all the things you did where you went to school, but she is currently with the Denver Museum of Nature and Science. And so let's welcome her, and then I'll introduce you. And from Eastern Michigan, we have Dr. Cara Shilington, and she's doing research on this particular population of spiders, and I guess we're probably going to hear a little bit about that
Yeah.
today, right?
Yeah.
So again, let's welcome them and say thank you for coming.
So thank you very much for coming. As he said, I'm Paula Cushing with the Denver Museum of Nature and Science. How many of you heard about this through the media? Raise your hand. Okay. So yeah, it's getting out there. So I am really here just as her backup band. So the main show is Cara Shilington because Cara and her team are doing the actual research down here. And what we've done in the past is we've talked a bit about spider biology, about why spiders in general are so important. But we're gonna switch things up a little bit today at today's lecture, and we're gonna talk about spiders as prey. Now we know, everybody here should know, that spiders are actually predators, major predators of insects worldwide. Go ahead. And so there was a paper that came out several years ago that estimated that the worldwide population is responsible for eating 400 to 800 million tons of insects every year. In the same paper, they estimated that the human population is only eating about half of that, about 200 million tons of protein. And of course, the media took those two factoids. I love the media, don't get me wrong. If you're media here, you're great. But they took those two bits of factoids, and the next day there were these headlines that said spiders could eat the human population, which of course is not true. But they're very important predators of insects worldwide. Next slide. But they are, and they're very abundant. So you'll find them. There's a myth that there's a spider within 10 feet of you. That's for sure true here, because we've got live spiders in the back. But spiders are found in every terrestrial ecosystem on Earth except Antarctica. And they are very, very abundant in these ecosystems. So if you go out late in the season when the little baby spiderlings are dispersing using a flying process called ballooning, you might see a field that's absolutely covered in spider silk. So that could be pretty close to accurate. Next slide. And there are, as of this week, there are 138 different families of spiders worldwide. There are over 4,000 different genera of spiders, and there are over 53,000 described species. And I say this week because that number is increasing all the time with colleagues of ours who are describing new species. It's estimated that there's another 300 to 700 species yet to be described in the US alone. They're found in all terrestrial habitats except Antarctica. They can get pretty large, and we'll see fairly big ones that are maybe 2 inches, the tarantulas that we have here. The really, really large tarantulas, very rare species, can get up to 30 centimeters, so about almost a foot. But that's uncommon. Most species are very small. And as small animals, that means that although they are top invertebrate predators, they are also prey for a lot of different organisms. So they are prey for a lot of different species of reptiles. If there are reptiles out there that will snatch up and eat arthropods, insects, they're going to also eat spiders. Hummingbirds have been seen to actually hover in front of an orb web and snatch the spider right out of the orb web, or snatch its wrapped up prey. So the hummingbirds and other birds that are insectivores, that eat insects, eat arthropods, will also readily eat spiders. There are mammals like this little kangaroo. Actually, kangaroo mice mostly eat seeds, but a lot of different mice, a lot of different rodents will readily eat insects and spiders if they can get them. Other spiders and other arachnids, like this scorpion is another kind of arachnid, and it will readily eat spiders, as will other spiders. And I think you'll learn a little bit about that particular risk as Cara talks today. So the spiders Oh, and there are also spiders that feed exclusively on other spiders. So there's a species called, in the genus Portia, and also spiders in the family Mimetidae, and those two groups will specialize on eating spiders. And Portia just has this fascinating behavior where it has incredible behavioral plasticity. It can change its behavior depending on the kind of spider it's hunting. When I was teaching at the college level, I shouldn't say this, but sometimes I would say that Portia was smarter than some of my students, because it would really be able to maneuver and manipulate to hunt a different kind of prey. Pirate spiders will hunt the other spiders. They'll specialize on them. Next slide. And the pelican spiders, this is a picture I took in Madagascar last summer. Pelican spiders are this is on our life list. Like, all arachnologists have pelican spiders on their life list. Birders are not the only ones that have life lists. Pelican spiders have this crazy, bizarre head. So this is its cephalothorax out on a neck with these very, very long jaws. And those are adaptations for them hunting other spiders, because spiders are predators. They're dangerous prey. So there's a lot of risks out there for spiders. And there's this animal, which, how many of you have seen this wasp? Pompilid. This is in the family Pompilidae. There we go. A beautiful wasp, huge wasp. It's also called a tarantula hawk wasp, because it will fly around and sting the spider in its nerve center. And we're going to learn a lot about this wasp in Cara's part of the talk. But it will sting the spider, it will paralyze the spider, it will then drag the spider into a burrow where it lays an egg on the spider. The egg hatches, the larval wasp will start to eat the paralyzed spider until it's finally dead. But it's an amazing life history, and again, we're going to hear about this much more. We also have mud daubers in the family Specidae. If you've ever seen mud cells, they are packed with paralyzed spiders. So specid wasps are doing the same thing. They're hunting spiders, and each wasp will sometimes become a specialist on a certain kind of spider. So you might open one of these mud cells and see nothing but jumping spiders, or open one of a different female wasp and see nothing but orb weavers. So they get a search image. When I was a young budding biologist, we had mud dauber nests on the side of our house, and because I was a budding biologist, I thought, well, this is interesting. What is this? And so I very carefully scraped it off the side of the house, and then very carefully opened up the mud cell and just about screamed, because my hand was filled with barely alive, wriggling, half-dead spiders that were all paralyzed. A little bit gross, but very, very interesting. Next slide. And they are also spiders, the other risks that spiders face. They face lots of risks. And I didn't even talk about the risk to being run over by cars, which the tarantulas here are at risk of getting run over by cars. But the spiders are also at risk to getting parasitized by different wasps that will lay their eggs on the spider. Next slide. And sometimes these eggs will hatch out, will develop into a larvae, and then the spider is still alive, still behaving sort of like normal, but that larvae begins to digest the spider's body, and it will oftentimes will lay their eggs inside the spider's body. So this shows you some pictures of some of these spiders that have been parasitized, where the parasitic animal has laid its egg on the spider, it's developed into a larvae, and the larvae has begun to feed on the still living spider. And next slide. So I was involved in an experiment that I'm not going to talk about today, but it was to send spiders up to the International Space Station. And one of the spiders that we sent up to the ISS was a wild-caught orb weaver from Florida. And we didn't realize it, but that poor spider not only had to figure out how to adapt to this bizarre near-zero gravity environment of space, which it did. It built a perfectly functional orb web. And it actually, we got the carcass back when it returned to Earth, and the woman I was working with at BioServe Space Technologies sent me this picture and said, okay, I see the spider up top, I see its legs, but what is this squiggly thing that's attached to it? And I said, I have no idea. So I got the carcass, I looked at it, and I realized that this poor spider had been parasitized by one of these parasitoids before it flew. So not only did it have to deal with near zero gravity and figure out this whole different habitat and environmental conditions, but then this parasite exploded out of its body, which couldn't but remind me of a certain scene, next slide, of a certain movie. So I did feel really sorry for this wild-caught spider. And some of these parasitoids will manipulate the behavior of the spider, where they don't kill the spider, but the spider is changing its web-building behavior to facilitate the cocoon building of the parasite, which is pretty fascinating. It's a fascinating adaptation. And these modified webs then that are triggered by that parasite are facilitating the cocoon structure and larval development of that parasite. Then you have kleptoparasites. You have things like this spider, which is a very shiny, it looks like it's covered in silver. It's called Argyrodes, and Argyrodes oftentimes hangs out on the webs. It's a very lazy spider. It hangs out on the webs of larger spiders. It does not bother to catch its own prey because it's living on somebody else's web, and instead it steals the food of the prey that the bigger spider is catching. So next slide. This is a picture. So the big spider there is a golden web-building spider in the family, or in subfamily Nephilinae family, it doesn't matter. It's in the orb-weaving spider family. That little spider is one of these little lazy spiders, Argyrodes, that's going to steal prey when the bigger spider catches prey. And spiders are also at risk. So I talked about all of these predators, parasites, parasitoids that are putting spiders at risk. Spiders also have to grow. So spiders have an outside exoskeleton, which means that as they develop towards maturity, the only way for them to get bigger is to molt out of their exoskeleton. And the new exoskeleton is developing underneath the old one, and as they hatch out, or not hatch out, as they emerge from that old exoskeleton, the new exoskeleton is soft, because that is the brief period of time when growth can occur. So the spider is very, very vulnerable during molting to predators. And that includes tarantulas. In fact, tarantulas have the bad luck, or good luck. They're more primitive, more earlier evolved lineage of spiders. And most spiders will stop molting when they reach maturity, but not tarantulas. So these guys that we see out there at this time of year, the females will molt throughout their whole lives, and their lives can last for 20 years or more. Whereas the males will molt until they mature because they just die. They just want sex, that's all they want. And then they'll die after that season. But during that molting period, it's very vulnerable. Spiders are very vulnerable against predators. So most spiders will molt in protected areas. They molt inside burrows, or if they're a web-building spider, they'll drop down on a silken cord away from the web, away from vegetation, and molt. So molting is a really high-risk period of time. This shows a tarantula pulling its new exoskeleton, its newly formed exoskeleton and body out of the molt. And you can see that even the fangs are not sclerotized, they're not hardened yet. So the whole body is pretty soft. Again, that's because it gives it some time to grow. And then, so spiders have evolved all sorts of adaptations, all sorts of ways to hide against all of these threats. And one of the ways they hide is through camouflage. So who can see the spiders? Can you see this? Let's go to the next slide. They're the spiders. But they're very camouflaged against their background. So a lot of spiders have evolved these color markings that allow them to become hidden against whatever background they're hanging out with or on. Some spiders have evolved ant mimicry. So ants, how many of you would willfully, gladly walk across an active ant nest? None of us would, right? You're a little crazy. And we don't do that, right? Because they're going to attack en masse. They can sting, they can spray acids. And so most arthropod predators that will readily eat insects, most of them avoid eating ants, and they avoid eating anything that looks like an ant. So many different species of spiders have evolved behavioral and morphological, anatomical adaptations that make them look just like ants. The plate on the left, those are all spiders, not ants. In this plate on the right, the middle three animals are all spiders, not ants. Next slide. And this mimicry can be exceptional. So again, it's where the thought is that these are Batesian mimics. The mimicry has evolved, the behavioral and the morphological mimicry has evolved to hide the spider from its own predators. It looks like an ant, it's not worth hunting. Next slide. I'm gonna skip that. Skip it. Another thing that spiders have evolved is this really interesting web decoration called stabilimenta. And the stabilimenta are these bright white bands of silk that some orb-weaving spiders decorate their webs with. So you can see a circular band of silk in the bottom right, kind of a linear band of silk, zigzag silk, and a cross band of zigzag silk on the upper right. And there's several hypotheses as to the function of these web decorations. One hypothesis is that it hides, these are spiders that hang out in the middle of their web during the afternoon when predators are active, and so that bright white silk hides the outline of the spider so it cannot be seen by a visually hunting predator. There's other hypotheses that suggest that the bright white silk actually attracts insects to the web, and it could be that it's functional in both of those cases. So there may be multiple ways that white stabilimenta or decoration is adaptive. And this is, I love this picture. This is one I took, I think, in Florida. And it looks like this spider web has two hubs and two spiders in it. The little knobby thing in the bottom hub, that's the spider in the bottom part. That's just junk above it. So in this case, the spider is using both the stabilimentum and junk in the web so that it's got at least a 50-50 chance against a visually hunting predator. Next slide. And now I'm going to turn it over. I have no idea what this is for. Okay. And actually, let me thank, before I turn it over, let me thank my crew who are in the back. I've got 4 volunteers, Karen, Katomi, Ian, and Jaden. We have an information table set up back there. We have a couple of live specimens that Cara has provided us that you can take a close look at after our lecture. So be sure to visit our table.
So thank you. So I am going to talk now, and we're going to come back to some of the things that Paula mentioned in terms of, what are the hazards or the challenges, and yep, there definitely is some death in this as well. So last year, I was very fortunate to be on sabbatical, so I actually spent 3.5 months in La Junta and worked out at the Comanche National Grassland. So I'm going to start with some of the information that I was tracking males during the mating season. So I'm going to start with some of the data that I collected there, and then also talk about some of the things that we are working on this year. So I'm going to start off with just some thank yous. So many of the photos that you will be seeing, including that very first one, were taken by Devin Matthews, and he is here again this year, so wonderful photos. Last year during my time here, I did have a field assistant, and so you will see her in the backdrop in many of my photos as well, and she did come out every single night. I was out every night at the field, and so this is Flora, and again, another photo taken by Devin. And then again, just a big group of people. The longer we've been here, just the more people we meet, and it has been just a wonderful community to work with. And again, the Tarantula Fest committee have been so supportive of all of our work, and I've had different people visiting me over the time that I have spent here. So just a reminder with the grasslands, so this is a huge area, and of course the important thing is that this is where we are finding a lot of tarantulas, and so hopefully you will all have your chance to be out there and see some of the tarantulas. So what I was doing last year was I was radio tagging the males, so following these males throughout the season and trying to get some ideas. So, you know, you're going to hear me say a lot, it's like, I don't know. We don't know a lot. We see these animals coming out. It's like, you know, how long have they been out? When did they first come out? What makes their decision to come out? Are they finding females? How many females do they find? You know, we don't know the answers to any of those questions. And I still don't know the answers, but I can tell you a little about what I did find during this period. And so I spent a lot of time following these animals around known burrows. So I knew where some of these burrows were, and then was looking to see how the males were moving. You can tell the time of year. This is hunting season, so Flora is now sporting her hunting jacket during hunting season. So some of the things that I was doing, again, just looking in close proximity to the females. So, you know, when I was out there and around these female burrows, if I found a male that was close to that burrow, I was looking to see what was he doing. So, you know, was he interacting? And so this was, you know, 108 of these males that I found. And within this area, some of them are just out there. I don't know how they're finding burrows. I'm trying to get to that question. But, you know, we see them within close proximity, and some of the times they're just sitting there. And, you know, we tested this last night. We're out there watching these males walking around, you know, and the burrow's like there, and then the male wanders in this direction. And it's like, we're, you know, trying to coax these things along to finding the burrows. It's really interesting watching them, and we still don't really know how they are finding those burrows. But I did find a bunch of them that were right at the entrance to the burrow. So then the next question is, there's lots of burrows out there. How many of them are actually females? And do the males actually know if they are females? Turns out maybe yes, maybe no. So, you know, so there were a bunch of them courting. And again, that courting isn't always successful. You know, there are sometimes juveniles, and these males will court juveniles. You know, I've seen very often if it's an unsuccessful, the male will be courting, and the burrow occupant, maybe a juvenile, sometimes it's another male that hasn't come out yet, they will flip over and expose their undersides and their fangs. And it's just a sign of disinterest. Go somewhere else. So I also had, you know, they would be at burrows that there was nobody in the burrow, or that the burrow was plugged. So, you know, they were finding any burrows that they could find. Interestingly, later in the season, we saw 2 cases where a male dug out the female. So she had put a plug of dirt on the entrance to that burrow, and we sat for about 2 hours and watch him painstakingly dig this out. He would lay web on the surface of the dirt and then turn around and pick it up with his fangs, cart it out, go back. Did this for almost 2 hours. Finally, the female did come out, and they did actually mate. Never seen anything like it. So, you know, again, another reason, be out in the field. You see just the coolest things. So one time we saw it was successful. The other time it wasn't. But, you know, sometimes I know that there's somebody in that burrow, and they just don't respond at all. And I don't necessarily know what that means, because the other thing is I can't tell who's in that burrow. You know, when they are small, you know, if it's a juvenile, I don't know if it's male or female. I don't know if they are sexually mature. There's just no easy way to know some of this. And so, you know, it's just, there's so much that we're still working on. So the 2 different ways that I was tracking these males is I had radio tags, and I'll show you some different data, and then I also have these little bee markers, and we're still doing this year. So expensive, very time consuming, very cheap, but you don't get a lot of data unless you see the animal again. So because I was able to be here for 3.5 months, I could be out with the radio tagging. So my tags last for about 28 days, and you have to keep up with the animals, otherwise you lose them. And you lose them even if you're keeping up with them. These ones, we just put these little bee markers on, and each bee marker is an individual marker. You put them back out at the site where they are, and you hope that you find them again sometime later in the season, and then you can kind of, how many days have they been out, where did I find them, things along those lines. Interesting. Okay, so the radio tagged male, so this is Everett, so there's a couple of people that are in my presentations that you'll see here, so Everett's been out in the field with me. So I release these males, the radio tagged ones, I follow them around, and the other ones just wherever I find them. Okay, and again, what I'm looking for is, when you release them, do they stay close? So these burrows, I think by the end of the season, over 300 burrows flagged. So I wanted to see, are the males staying within this area, or are they moving beyond this area? Just some idea. I need a starting point here. And then, when I'm out there and I'm following them around and I see them, what are they doing within this area? How often are they finding these females? So the other thing that after being out, trying to divide this time period up into, what early season, middle season, late season. And so it turns out that it was easy to do it by rain events. So the tarantulas have a hydrostatic skeleton, which means that they really need water for the limb extension. And so when it's really dry, they start to lose water, and it really impacts their ability to move. So one of the things that I looked at was dividing this mating season into rainy periods. And so, so you see these 3 periods here, early, middle, and late. You get some idea of the amount of rain that was occurring. But really, for especially this middle part here, 26 days with no rain out here. So this is really a tough environment for these organisms. They don't have a permanent burrow. Those that are in burrows can go into their burrows. They have some humidity in their burrows. These guys, this is a really harsh environment. And so I was interested to see, do we see differences across the year because of some of these environmental factors? And we're kind of doing the same this year. So this year we have been out. We came out for a week in August. We're out now, and then we're going to be out a week in October. And same thing, we're kind of looking to see what are some of the changes that we see across this period. And so this gives you some idea of the yellow down here, these are the numbers of males that I found on any particular day. So, we're kind of within this region here, so we're sort of at that peak of the season. I am finding them throughout the season, but around in mid to end of September is when we are seeing more of them every single day. And then we also see some of the temperature and the rain events. So the humidity is in blue. So again, we can see with those rain events coming, we have really high humidity, and this seems to be important for the animals. And with the high humidity, we also see a drop in temperature. So both temperature and humidity is really important for determining when these males are going to be active. The other thing that you see with these major rain events is this is what my animals look like after it has rained. So they are covered in mud. So some of these will find refuges. They'll go into any types of little burrows, but you can see that they're covered in mud. So the water will just run into those burrows. This is also the time where you see a lot of them, readjusting their homes. So there'll be a lot of digging. They'll reorganize those burrows after the rain, because the rest of the time the soil is just too hard to dig in. So we can clearly see that these events are really impacting the animals. This is the end of my field season. November last year, we got just incredible, how much? 3 to 4 feet of snow, literally in 24 hours. 2 days before that, Sue and I had been out at the field site, and we had found 3-4 of the B-market males, and then this came. I couldn't even get out to my field site. I couldn't leave town. I had to ask my Airbnb person if I could stay a couple of extra days because we were completely snowed in. And so again, just thinking of these impacts. So, any males that were still around at that time, after this, I'm pretty certain nobody was around after this. They don't have burrows to go to, and so many of them will just die with the cold as well. And so this gives you some idea of, again, I've got these periods. This is divided, the first, the middle, and the end. And so some of the data here, looking at distances traveled, one of the things that is interesting, and I'm still working with this, there is smaller distances traveled early in the season. The numbers above each of these are the number of days that I have them tracked. So it turns out radio tagging is really difficult. Very often, the animals die. Some of the times, you just arrive and the animal's dead. Don't necessarily know why. Other times, they do disappear into a female burrow. Not a huge number, but some that does happen. And then other times, I just, the day before the signal was there, I go out the next day and the signal's gone. So I have no information as to what happened with these animals. But you notice that early in the season, these animals, even being out 19 days, 18 days, they're hardly moving at all, which is crazy to me. This is the mating season. Shouldn't you be out looking for these females? Maybe not early in the season. Then later in the season, we start to see much higher levels of activity. So again, this, didn't know that, and so, some different things to think about. I'm assuming that, many of these 28 days, my tags are only, that's as long as they last, so probably many of these, the tag just ran out, so the male may still have been alive at that point, but I just wasn't able to find it at that point. And then I had a couple that, I immediately put them out and they were dead within one day. So it's tough for these males being out there. Some of the times I know what happens with them, but not always. This is looking at my bee market males. So I had 279 males that had an individual bee marker on them. Captured them and then released them out at the site that I captured them. And again, you can see a lot of them I am finding again, within a day or two after I release them. So they are staying, at least for a short period, relatively close to the area that I released them. But some of them, were out for 27 days. And so we also were finding some of them on the very last day. I'm out there every single day. And then I don't see them until the last day. It's like, what are they doing in between this time? Why am I not seeing them? And again, there's no way to know. I can only know when I collect that information. So it's great to do this and you get some really interesting information, but there were only 42 unique recaptures. So out of 273, it's a whole bunch that just have no idea where they were going. And again, this is just some of the bee market males. So, that the first time when you see the tarantula, it's like, does it have a bee marker on? So I have lots of pictures of these males when I found them again. It was really exciting to see them with their bee markers. The other thing that was interesting, so coming back to this, the challenges that they're facing is missing limbs. I was expecting more for these animals, but actually just a very small percentage. So out of that 273, only about 5% of them were missing at least one limb. There's some really interesting studies looking at handedness, as whether you're right or left-handed. There are some studies that show that some spiders are handed. They do have a right or a left preference. So far, I'm not seeing anything with the tarantulas. And the other thing that we're seeing is it is most common, even though I'm showing one of the fourth legs back here, most commonly they are missing the front pair of appendages and the first legs. And I do think that probably these losses are due to interactions with females. But again, I never actually saw any of this. This is, again, even with the radio tags, it's, unless you are there when an interaction is happening, you're still not able to see a lot of what is happening. So that's just my presumption. And then, the types of things. So this is a male that I did not tag. It had nothing on it, and there it is dead. Don't know why. There isn't really any sign that something has eaten it. Maybe it was, it overheated. Again, so many questions. This one was one at the end of the season. So this one just literally wore himself out. He was one of these ones that was, in those last few days, was really walking huge distances. There weren't many females out, and he was still out there trying, and just, within, a couple of days, he just got slower and slower, and then the last day I came out, was just no longer living. And then this year, so we're out for just these really brief periods, and so we came out in August, and we're looking for male tarantulas crossing the road, and this is what we found. Every single tarantula that we found for the first couple of days was accompanied by a wasp and was paralyzed. Every single one. How crazy is that? Again, I was out all of last year. I maybe saw over that whole season, 4 or 5. And it'd be driving down the road. Oh, there's a tarantula. Oh, look, it's another paralyzed one. Oh, there's a... No, you look at my logbook. It's like every couple of minutes I have a tarantula, but it's paralyzed. So once again, I spent a fair amount of time looking at the wasps. So the wasps, when I'm seeing them with the tarantulas, typically they will drag these tarantulas by their fangs and drive. And then the other thing, they're dragging them down the road. They're not going off to the side of the road. They're going down the road. Every one of these was down the road. We have a field site with hundreds of burrows. Were there any tarantulas paralyzed there? No. They're all on the road. So are they dragging them from the burrows, bringing them to the road, and then taking them down the road because it's easy to drag them down the road? I don't know. Just, again, more questions. Really interesting to think about. So most of the time they'll be dragging them for a bit, and then this species, I think, we're still looking at some of the data, they look for a burrow to bury them in. Some species will build a burrow and they take the tarantula and bury them in that burrow that they built. I think this species drags, and so then they'll let them go for a bit, and then they'll start circling, and I think they're looking for a burrow. So while they were doing the circling, I'd come in, grab the tarantula, and so I did collect a bunch. This one stared me down. I was never able to get the tarantula away from this one. And I'm usually okay with things. This thing, every time I approached her, and she's eyeballing me, I'm not kidding. And I'd go down and I actually scuffed dirt at her at some point. She would not leave. And this was a really big tarantula. Would not leave that tarantula. So again, just some of the differences in these interactions was just amazing. So she will have a baby. The other ones that I stole the tarantulas from, they will not, unless they find another tarantulas, they will not have babies. So these are all of my paralyzed tarantulas. So we took them back to the lab. We have them all lying on their backs, and we're giving them water because the biggest concern is that they dehydrate. These animals can go really long periods without food, but I mentioned that hydrostatic skeleton. If they don't have water, this can be a real major factor for them. So we have them all on their backs, and every now and again we are giving them some water. This is Everett helping us package these. So normally when I ship tarantulas back, you put tarantulas in a container and they can support themselves. So if the container moves, they've got their legs out to the side, we've got some padding in there, but they're okay. These guys are not okay. If that container shifts, they can really get shaken around in those containers. So we spent several hours carefully packaging, putting that cotton balls and paper towels to really make sure that they were protected in the shipping, sending them back to Michigan with us. And at our lab, so this has been about a month that we have had them in the lab. They are starting to recover from this. So I was just talking to Warren, and we're trying to, think about what do we know about this venom. So it's possible this venom degrades. And so what I have up top here, one of the things that I'll talk in a little bit about my current grad student, we're measuring metabolic rates. And for these males, we're looking for transitions over the mating season as this is happening. So up at the top there, you're looking at literally CO2 production. So when we breathe out, we're breathing out CO2. I have a machine that can measure it. And so you can see in the top, my scale This is parts per million of CO2. You can see all of that up and down. These are the males that are active in these chambers when I am measuring them. So they're around about, up to 400 parts per million. These are my paralyzed individuals. The scale here, the highest one is close to 70, but they're not doing anything. But this one, I think, is starting to recover. So we're starting to see some movement. So we can measure some of these differences as they're coming out of that paralysis with these metabolic rates as well. And we can definitely, now all of them, we've actually righted them. Many of them are supporting their abdomens, and they're not necessarily walking around, but there are clear progression with them coming out of this paralyzed state. So I'll be away for back in a week, and we need to start making some decisions. How do we feed them? They're not going to be able to grab food. We're going to be doing crushed crickets. I don't know. It's a lot of care for these animals, but they definitely are coming out. So this is, again, wonderful photos by Devin. This is my grad student this year, Spencer. I saw him somewhere. So if you have tarantula questions, he is another good person to be asking. And he is really focusing on looking at some of the metabolism of these tarantulas as they are dying. And so he can tell you a lot about this process of what is happening physiologically with these animals. It's a programmed death. We can keep them in the lab. We can give them all the food that they want, all the water. They're kept at nice, warm temperatures, and they will die. There is nothing that we can do to stop that programmed death. And so he's actually measuring the progression of this. And so we're out here collecting some of these animals. And we are also, we call this a racetrack, it isn't. We're looking at just locomotion. So how does the locomotion change? As I mentioned, when it's really dry, these animals are noticeably wobbly. Does the speed change? I mentioned also at the end of the season, they're actually walking further. So is there an increase in their speed, or are they just time? It's just a matter of they walking for longer periods of time. So we have this little racetrack here. There's a camera. We send our little tarantula in, and we're just looking at voluntary locomotion. So we just kind of start them. We've got a little paintbrush just to give them a little prod to get them to move, and then we're just recording how long it takes them to move a certain distance, and we'll get some idea of speed. And we're doing this over these periods, and then we'll do something similar in the lab to see the progression. And and then we can start to think, what does this mean for their ability to walk around and find females in their environment as well? So sorry for the abrupt entrance, but that was as far as I got. There was supposed to be a really cool photo that Spencer took, and I just didn't upload it. But that's kind of where we are. We just got out of the field this morning. We were releasing some of our males that we had captured, and so we are just always so happy that you are here and interested in the tarantulas, and we would be happy to answer any questions.
You kind of spoke to it, but do you get any sense that they navigate, the males, or are they just on a random walk? And if they navigate, how do they do it? Because they can't see horses.
Right. So do the tarantulas navigate, the males in particular, when they are looking for the females? Absolutely not. I have. In Texas, and I haven't done it recently, but just based in Texas. So in Texas, I was tagging some males, but I also spent all night with some males, and I literally followed behind them with flags. And every couple of minutes, I would put a flag down. And it's pitch black out there. I have no idea, but I've got numbered flags. And so, you're just wandering randomly at night. And then I come back during the day, and I kind of look at the locations and where, I've written down which time I set every flag. I came back the next morning. It was circles. I had been walking circles, and I hadn't even gone that far. I mean, it was just absolutely crazy. And so, the same thing. Every now and again, I think, oh, he's got a good idea, and he's heading down one way. Sometimes it is roads. Some of the males that move the furthest would sometimes hit a road. And, I think these roads are these corridors, and they just kind of get going. But the rest of the time, it feels just all over the place. And, it's just, with spiders, what do you know with pheromones? I mean, what do you know about finding females with pheromones?
Yeah, so it's thought that there are some species where the females will lay sexual pheromones in the silk. And it could be that that's what's going on with these.
So I don't think they're doing that. Again, Devin and I were out last night, and I'm not kidding. The burrow's right there. You know, you just want to tap this guy, go, just turn that way. You know, why are there no pheromones out there when they're that close to a burrow? So interesting that, you know, that we still know so little about this one.
So evolution is not selecting for smart males, is what she's saying.
Do they always circle around? Like, do they keep on circling and it gets bigger, or do they just randomly?
No, it's absolutely random. Yeah, so I had one of my grad students did do the stats, and he did, and it does turn out to be complete. You know, I say circling, but when you look at it, it actually isn't circling. It is completely random. It feels crazy.
I know your focus is on tarantulas, but has anybody done a study on the predatory wasp and their populations?
Do you know about the predatory wasps?
What was the question?
About the predatory wasps that prey on them, and has anybody studied them and their populations?
So the question was whether anybody studied the pompilids, the tarantula hawk wasps, and I don't know that. So that's literature that I don't keep up with. I would assume that there's something known about the basic biology. There was a biologist named Crombine years ago who produced a very large manual of trap nesting wasps. And I'm almost certain he's got some information about natural history, but what that would be, I don't know.
Yeah, and I know Warren, where's Warren? He was looking at some papers too. So he's another person, you know, this is just kind of a new thing that we're looking at, and so he's been looking at some of the papers with some of the venom, what it means. Spencer and I did just a quick search, and there's not much. You know, we were finding this activity was mostly occurring between 11 and 1. So I think during the day it's too hot. Both the wasp and the tarantula will, you know, if it's a really warm day, they will essentially cook, especially a paralyzed tarantula can't get out of the heat. But there's not a lot out there. And I watched one for an hour and a half. And I've got the GPS coordinates of, you know, how much it was moving every time. And it's just, this down the road thing is just really something that I'm interested in. And I haven't seen that in any of the publications. And we haven't done a very good search, but we are really interested now in looking at some of these.
So where else in North America and around the world do tarantulas live? And what are the mating seasons like in North America?
So the mating seasons of tarantulas, so once again, what a great question. Do we know? No. So there are colleagues in, there's a big tarantula group in Uruguay, and there is a big group in Argentina area, and there's a woman out there that does phenomenal work, actually similar to what I do. So those are areas where there is some information known. I've also got a colleague that works in Mexico, so we know a little bit about the species there. In North America, you know, we have some colleagues that know where to find them, but, you know, other than just their natural history, there is just nothing. And, you know, the interesting thing also with this species, we're at the northernmost edge, and this is one of the most widely spread species. You know, so it's really interesting. Is what we're seeing here, is it the same somewhere else? When I worked and did my tagging in Texas, it was a different species. Their mating season was in May, June. So it's not at the same time of year. So I don't even know if within this species, whether we can necessarily expect the mating season to be the same. You have anything to add? Yeah, so it's, yeah.
So we were out there yesterday and came across quite a few burrows with an empty egg sac. Is that this year's, last year's, or do they have multiple litters?
Oh, so great question. If you see egg sacs, so we are seeing egg sacs, and this is actually one of the ways that we are trying to sex these animals. If I see an egg sac, the burrow is occupied. So the easy answer is you need to feel the egg sac. So this year's egg sacs should have been kicked out of the burrows literally probably within the last month. And so if you feel them, they will actually feel pretty soft and silky. If it's from last year, it's usually very flattened. And if you kind of peel it off the ground, whereas this year's I'm seeing a lot more softness to them. And that's as much as I'm going off of as saying, but you will sometimes see some from a couple of years ago. But yeah, they just recently had their babies.